All right, everyone. So today, what we're going to be talking about, or today, again, today's lecture, we do not have it in person, but I want everyone to review all this stuff for next week. Um, so yeah, so we can talk about it next week and we don't seem like we miss a week. So first one we're going to start with is cellular transport. Now, we didn't get into it last week, but we'll talk about it here. I hope everyone can hear me because I just the first time doing this. I hope everyone can hear me. Okay. So when we talk about cellular transport, we're talking about the movement of molecules coming in and out of cells, specifically here. Um, it all has to do with this idea of Brownian movement and how molecules will interact with each other. Okay, so the definition, again on the screen, it says the erratic random movement of microscopic particles of a fluid and a gas. Now, if everyone remembers in chapter 1 of lecture, whoever you had, if it's me or if it's someone else, you guys talked about um, the three states of matter. Okay, we talked about solid, liquid, and gas. Now we should remember how particles move in each of those types of, or in those mediums. So remember, a solid is definite volume, definite shape. The particles are moving much slower than a gas or a liquid. Now, when we have fluid, again, there is a definite volume, but no definite shape. The particles are able to move around and be able to conform to their container. And we look at those particles as being more fluid, allowing for more movement. And then gas is the most, okay, the particles are moving the fastest, they're moving around the most, they're constantly expanding to whatever environment that they are in, shape, and the amount of volume that they can have within that given container. Now, remember, if we heat or if we increase the pressure within any of these fluids or gases, we're going to be talking about an increase in the particle movement, which will increase of how things are being absorbed or how things are going to pass through membranes as well. Okay, the continuous bombardment of molecules in the surrounding medium, and again, increase with temperature like we just talked about, or like I just talked about. Okay, permeability, again, we talked about this in lecture. A lot of what we're going to talk about in this PowerPoint is going to be lecture stuff, but there is going to be some things we need to know about lab that I will talk about towards the end. Okay. This allows nutrients to enter but keeps other substances and regulates passage. So this is like a gatekeeper. What can pass through, whether it be very small or whether it have a proper protein, will be able to pass through a membrane or pass through that membrane. The big thing here is we're trying to keep proteins within the cell and we're trying to allow waste and particles out of the cell. Okay, there's two types of transport that occurs, passive and active. Okay, passive transport is diffusion, or the idea of concentrations going down their gradient, or down from high to low concentration, or high to low pressure. That's what's going to drive movement. Okay. We're always, like we talked about in lecture, or whoever you had in lecture, they should have talked about, when everyone sits in a room, we don't always want to huddle right next to each other. We want to evenly space ourselves out within that room. That's the same idea that's going on here with diffusion, is we want an equal concentration or an equal spacing of molecules within a medium where everyone's happy. Okay, we don't all in class huddle in a corner like penguins because we want to do that. No, that doesn't make sense. Okay, we're going to be evenly spread out as much as we possibly can. Down here, okay, kinetic energy, right? That's the energy of movement or the energy of molecules themselves. That's what's going to be the driving force of how fast diffusion occurs. Now, under normal circumstances, diffusion will occur normally, but the rate at which it happens is all going to be dependent upon the amount of energy within that system or how fast those molecules move themselves. So if you think about it, molasses is going to move a lot slower. Okay, the molecules are much tightly packed. It's very viscous. It's very solid than if we have liquid water. Okay, same idea here is that liquid water has more energy. The molecules are able to move around faster than something like molasses. Now, when we talk about solutions, a solution is the term used for what is being in a beaker where we have two components of it, a solute and a solvent. Now, solvent is always doing the dissolving. Water is the universal solvent, and solute is whatever is being dissolved. So when we talk about salt water, the solute, sodium chloride, okay, and the solvent is water. Now, again, the ionic bonding that we talked about in lecture, just a quick review, okay, when we have the dissociation of sodium and chloride through the ionic bonding, the charged particles will go through there, and again, this is the idea of electrolytes in a solution where the solute, sodium chloride, the solvent is water. Now, osmosis is the same thing that we talked about with diffusion, except it's the flow of water across a semi-permeable membrane. So here what we have is we have some sort of barrier that will only allow water through, and it will not allow any of the solutes through. In this case, it's sucrose, which is a sugar. So again, we want to have equal concentrations of water and salt. The solution wants to have equal concentrations on both sides. Okay, so the solute and solvent ratio wants to be equal. So here we're going to have much more water pass through this membrane in order to dilute the sucrose molecules so that we can at least match as much as we possibly can to this other side. Okay. If solutes can diffuse, water will move with them, and it's 100% correct. All right. We're going to skip that one. Okay, passive transport. Passive transport is the process of no energy being used for molecules to go down their concentration gradient. We have simple diffusion. Again, this is where we're talking about very small ions, very small lipid-soluble particles, because again, what's going on here? This is the nonpolar region of the cell membrane, and we have the polar region, the extracellular fluid, and the intracellular fluid, or the cytoplasm, is all water-based, so it's water-loving. So the only thing that can pass through here and go through this nonpolar barrier is lipid-soluble solutes, because again, lipids are usually not dissolvable in water. They're able to pass through and go down their concentration gradient. Now, carrier-mediated facilitated diffusion, this uses a specific protein in order for some sort of solute to pass across the membrane. Think about this as like a key, where we're trying to enter a room, but we have to have a specific key to get into that room. Okay, that's what's going on here. That carrier-mediated facilitated diffusion is like the door in which we need to have the specific key in order to get across that barrier into the room from the hallway into the room. Okay, channel mediated, this is very similar to like a hallway. Let's think about when we're talking about this, we want to, whatever can fit down that hallway will go down that hallway. Whatever can fit in that room or fit through that spot will be able to pass through. It's not specific to whatever, like over here, it's very specific to the solute being dissolved. This is doesn't matter what as long as it fits. Okay, and then an aquaporin is the same thing, but it's just for water molecules going down their concentration gradient. Okay, this is the big thing that I want you guys to know here. Say when we talk about solutions, okay, again, a solution is a solute and solvent. Okay, the solvent is water, the solute is going to be whatever's being dissolved. Okay, so here when we look at this, what we're really trying to look at is this is a cell in a solution. Now, normally we should have equal amounts of solutes and solvent to solutes and solvent. Think about that permeable membrane as the cell membrane of this red blood cell. Okay. Now, again, we're going to want, water is going to want to go to equal out the concentration gradients. So in an isotonic solution, we have an equal number of solute and solvent on the outside of the cell as it is to the inside of the cell. Okay. Hypertonic solutions, this is where we have a hypertonic solution. This is where we have a higher concentration of solute inside the cell. Okay. There's a higher concentration of solute inside the cell. Sorry. We have a higher concentration of solutes outside the cell in a hypertonic solution. That means it's extra salty on the outside. Hyper, think about extra outside the cell. And what's going to happen is water is going to flow out of the cell and it's going to start to shrivel and undergo this process of crenation. So when there is a higher number of solute outside the cell than there is inside the cell, water will cross the cell membrane and leave the cell in order to try to equal out the concentration outside the cell as it is inside the cell. Okay. And then hypotonic solution is the opposite. We have a higher concentration of solute inside the cell than we do outside the cell. So what's going to happen is water is going to rush in and that cell will swell. That cell will swell. Okay. That cell will gain in size. Okay. And it will eventually go through a process. If it gets too big and it's not able to withstand the amount of water coming into the cell, the process of hemolysis, okay, where the red blood cell explodes. Okay. Filtration is all, last thing here, is all dependent on hydrostatic pressure and all dependent on um, the amount of force that's being able to be applied through the membrane. Okay, this is what separates large particles from smaller particles. Okay, smaller particles will be able to pass through, filtrate. Okay, where larger molecules will get trapped and will stay inside, just like your coffee filter will stay inside. The grounds will stay inside coffee filter and just coffee water will come out. Okay, so that's what's going on there with cellular transport. The next, the next one we want to talk about. Okay, now we're going to get into histology. All right, we're going to talk about epithelial tissue. When we talk about epithelial tissue, or we talk about histology in general, right? This is the study of tissue in general. That's what histology means. Okay. We have four basic types. We have epithelium or epithelia. We have connective tissue, we have muscular tissue, and we have nervous tissue. Okay. When we boil down everything in terms of all tissues, we can be broken down into these four categories. Okay, epithelia has to do with skin, hair, nails. Connective tissue has to do with everything connecting organs, connecting different body parts together. This is cartilage, bones, adipose tissue, areolar tissue, so on and so forth. Muscle tissue, again, we have three different types we'll talk about a little bit later, and nervous tissue. Okay. Epithelium, where is that found? It's covering external body surfaces like the skin, lines tubules and cavities. We also have certain glands that make up endocrine and exocrine functions. Okay. Generally separates from our insiders or outsides, just like we talked about in the tegumentary system. Okay. Here, the polarity, or what we call the apical surface. The apical surface is the free surface in which we all name all cells and all epithelia based on. Okay, we're always looking at the top layer. The connective tissue basement membrane, or the connective tissue starting where all cells of epithelia start from, is called the basement membrane. And what we're talking about here is this is where we have vascularity. This is where mitosis is occurring when we're talking about epithelia specifically. And as we get further and further towards the apical surface, we have less and less blood supply going there. But the main blood supply is always supported through the basement membrane. Okay. Now, classification and how we describe epithelia is going to be based on two things. We're based on cell arrangement as well as cell shape. Okay, we have simple and stratified, where simple is a singular layer. There's only one layer of cells. In stratified, we have multiple layers of cells, more than one. Okay, when the way I describe this as is when we're naming cells or when we name a house, we don't name the house based on the first floor. We always name it based on the top floor. Okay, we're always naming it based on the apical surface, which is the top layer, and that's how we tell if it's stratified or simple. Okay, then we go based on shape. So here, shape, we have squamous, which I describe as flat plates. Okay, cuboidal, which look like cubes, and columnar, which look like columns. Now we're always again basing on the top layer, which we'll talk about in a second. Okay, we'll go back to that. So simple, again, we're naming based on the top layer. There's only one layer here, so this is simple, and they look like flat plates. This is simple squamous. Okay, here in stratified, again, this is the basement membrane or the basal lamina, which is where all the cells start from in this tissue. But we do not want to name it based on this bottom layer. We always want to name it based on the top apical surface. Okay, we're always naming it based on the top. So this is multiple cells, multiple layers of cells, which is the stratified type. And then there again, we always look at the top, squamous cells. Again, here these look like cubes, but we're never naming based on the bottom. We always name based on the top. Okay, columnar again, same thing here. We're looking at columns, and there's multiple layers. Cuboidal, there are two layers of cubes, multiple layers, and they look like cubes. Um, Again, and then up here, simple cuboidal, one layer, simple columnar, one layer. Okay, if we go back, pseudostratified, we have two different other types of epithelia. So stratified, cuboidal, and simple, and squamous are all of the three normal types, I would say. Then we have two additional types, which is pseudostratified. This is where there's varying height of the cells, but they all touch the basement, or they all touch the bottom, and they all touch the top. There's only one layer, but they vary in height. And the nuclei, this is very important, are always at different levels. When you're looking at a slide under a microscope, you want to look here. Often ciliated. This we'll talk about in a little bit. Transitional, this is where cells respond to stretch or distension. Cells will flatten when stretched and then rounded when empty. And I'll explain this maybe a little more in class when I can show everyone. Okay? Again, here, transitional all over the place. They do not all touch the bottom and the top. Pseudostratified, all cells touch the bottom as well as touch the top, as you can see here with my cursor. Okay, so here's some examples. Simple squamous, this is a single layer. Simple, single. Squamous is flat. Okay, so here we see that. This is the lining of a lung, the alveoli. Okay, we have a cell right here, cell right here. This is one singular row of flat squamous cells. Same thing going on here. Okay, stratified. Again, this is the basement membrane. We're not looking down here. We're looking at the top. As we can see, these all look like flat plates. Cell, 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 right on top of each other. Okay, and they're all multiple layers. Keratinized, non-keratinized, two differences. Keratin is waterproofing. We see keratinized structure found in the epidermis, which is on our skin, which helps waterproof our cells in general. Simple cuboidal. Okay, again here, let's look at this lumen. This is one layer of a singular circle. Okay, one layer with an inner lumen here. Stratified cuboidal. Here we should have two layers. This one actually looks like three. This part over here, two. Looks like we have a little bit more over here, but we have two layers. This is a lumen. This is the center of some sort of gland with cube-like cells. Same thing over here. One layer. As you can look, all the nuclei are in a row. That's very, very important. Columnar, single layer. Again, same thing here, single layer. These are usually found in the digestive tract. Stomach, intestine, gallbladder. Okay. Stratified, multiple layers. One layer here, one layer here. Everyone see with the cursor at the top left, we have two layers of cell nuclei, and that's the biggest way to identify what we have. Again, right here, two layers, much easier to see if you can follow my cursor. You can see my cursor. It's over here on the bottom and on the top. Transitional cells are all over the place, as you can see with my cursor. Okay, and same thing over here. Pseudostratified. Everyone look here at the nuclei. The nuclei, as we can see, they're not in a singular row. We have some in the middle, some at the bottom, some through here, ciliated. Okay. Same thing here. One here, nuclei here, nuclei here, 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 here. They're all over the place as we look at them through there. Okay. So if we look at this right here, everyone give yourself a second to take a look. Okay. We see squamous, so that should be easy. This we should know is simple squamous. Extremely straightforward. Right here, what do we have here? Okay, singular lumen, singular layer, and these all look like cubes. Transitional epithelia here, kind of all over the place. They're going to respond to distension. So when they're stretched, they flatten out. And when they're relaxed, they're bulbous. Okay, here we have, here this looks like transitional as well. As you can see, the nuclei are all over the place. Right here, stratified squamous. Right here, pseudostratified. Nuclei are all over the place. Right here, simple cuboidal. Right here, simple columnar. And last one right here, we have simple squamous. Okay, do these last on your own, and we'll check back in lab and go from there. Okay, so what we're going to go over now, the next lecture is going to be epithelial tissue. Not epithelial, muscle and nervous tissue. All right. Okay, perfect. Histology, muscle tissue. Nervous tissue is extremely easy, guys. When we're looking at these under a microscope, okay, we want to be able to identify this. This is the soma, or the cell body. Okay, we have an axon, and we have some dendrites coming out here. And we have the nucleus right in the center. Extremely important, extremely easy. Kind of looks like a little spider web through here that we're looking at these. When we're talking about it in lecture, we're going to go over the functions of all these, but I need you guys to know that memorize these. All right, take a look. Look at how the shape is. Look at how it looks like a giant kind of spider web with the center. Okay, and you'll have a distinct nuclei in the middle. That's going to be key. Muscle tissue. What we have here is we have three types: skeletal, cardiac, and smooth. Smooth muscle tissue, how we know it, it's non-striated. There's a singular nucleus per cell, and it's involuntary, which is what we're going to talk about in the muscular chapter. Okay, but naming the histology, non-striated, one nucleus. Cardiac, involuntary. Okay, so we can't control it. The heart beats on itself. Otherwise, if we had to think about it, we'd probably die very quickly. Cells are striated, so we'll show the striations. There's branches, they're connected, and they look like forks, and they're connected by intercalated discs. And then last one, skeletal muscles, they're voluntary. We can control them. They're striations and striated, as well as multinucleated. Okay, skeletal muscles right here. See the little ribs that comes through here? Okay, so when we look at that, all right, we're looking at striations, multinucleated, bunch of different nuclei in here. Okay. Cardiac, see the branches coming through here. See these little lines right here? These are intercalated discs that connect two different cells together as they branch. Multinucleated, and again, these are involuntary. So if you see these little lines through here, we want to know, or we want to be thinking cardiac. Smooth muscle, exactly as it sounds. There's no striations and it's smooth. Single nucleus per cell, single nucleus drawn out through that cell, through that cell. Okay, very, very important. Okay, let's name these. Very simple. This should be nervous tissue. Everyone should have got that right. Okay, down here at the bottom, we have smooth muscle tissue. We need to make sure that we spell it all out on the quizzes and tests. Smooth muscle tissue. Right through here, skeletal, multiple striations, and these should give away the intercalated discs, as well as the branched and multinucleated structures. Okay. Now the last one that we're going to talk about is connective tissue. This is the one that trips a lot of people up. All right, everyone, connective tissue. Okay, last thing we're going to talk about today, and we're going to do a lot of this specifically next class. Okay, so here, this is simply what we're trying to do is we're just trying to recall and name what we're looking at. So connective tissue in general, connective tissue is what we call anything that's connecting two structures together. And this could be part of the dermis, This could be cartilage, this could be bone, so on and so forth. So here, first one, at the end of this PowerPoint is the key. So that's a good way to study, and most of this is just going to be memorization. But when we're here looking at elastic fibers, we can see the orientation and how wavy everything is. Okay. Here, where it's not as compact, there's a lot of air. When we think about elastic cartilage or elastic connective tissue, okay, we're thinking about the nose, the ear, we're thinking about all that kind of stuff. Here, number two, this is adipose tissue, cross section. Adipose tissue is fat. So we should see, again, fat cells have a large amount of air or a large amount of space between them. So all these white marks are adipose tissue. This is often confused with the slides for simple squamous epithelium. So just make sure that you're really looking. Simple squamous, how we tell it's different is there'll be a lot more nuclei. As you can see, there's only one nuclei here, and there's only one nuclei per cell, not a bunch of nuclei wrapping around the cell. Okay. Here, this is reticular connective tissue coming from a lymph node. Okay. Again, we just need to take a look at how dense and compacted everything is through here. A lot of this, again, mostly muscle memory of memorization. Okay. Number four, this type of connective tissue is blood. Most people forget about it. All these little red dots or pink dots in here, red blood cells, and then the purple stained. What that is that is going to be white blood cells or lymphocytes. Okay, connective tissue through here. This one, this is elastic cartilage. Number six through here, these are chondrocytes and hyaline cartilage. Okay, this is found at the end of bones or through endochondral ossification. Seven, okay, what we're looking at here, this is connective tissue, of course. This is compact bone. We should be able to see the osteon. We should see the osteocytes and the lacuna, okay, that we'll talk about as we get into chapter 6. Right here, again, we're back. This is adipose tissue. Again, look, one cell, one cell, one cell. This is not simple squamous epithelium because we would see a bunch of cells wrapped around. Okay, connective tissue through here, number 9. What we're looking at here is looking at some dense irregular. As you can see, the fiber orientation is all over the place. We'll talk about when we get to the integumentary system where we find all this type of good stuff. Number 10, this is spongy bone. Okay, so spongy bone in the trabeculation is found through here. It's found in the metaphysis and diaphysis of metaphysis and epiphysis of the long bone, mainly. Spongy bone. Okay, number 11 here. The way that the fibers are oriented, everyone should see dense, regular connective tissue, how compacted everything is through here, and how everything is in a linear straight line. Okay, this is not irregular, like we talked about here, how everything is all over the place. Right here, this is dense, regular connective tissue. Okay, and make sure that when you read the questions on your quizzes and tests that you actually are looking, again, at fiber orientation and what the question is asking. All right, 12 through here, this is dense. No, this is areolar connective tissue. Okay, areolar connective tissue. Again, this can often be confused with nervous tissue. Don't get them confused. Make sure we're looking at what type of connective tissue we're talking about, not what type of tissue in general we're talking about. Okay. Through here, number 13, again, here we have elastic cartilage. We went over one with elastic cartilage a couple slides ago. Again, here, elastic cartilage, what we're looking at is we're looking at the orientation of the chondrocytes and how this can be able to bend. Number 14, elastic cartilage, elastic connective tissue, I'm sorry. Elastic connective tissue through here. Look at the orientation, look how wavy it is. This is very bendable. 15, okay, 15 here. What we're looking at here, this is spongy bone. Okay, when we're looking here at spongy bone, okay, we're looking at the ends of the long bones. Again, all memorization. We're going to talk more about this next class. 16, okay, look at the orientation. This is dense regular connective tissue. Okay, look at how the orientation is. We're in a linear straight line. 17, okay, we're back to adipose tissue again. Another example. Again, one nuclei around the entire cell. 18, okay, look at the fiber orientation that we talked about. Okay, it's all over the place. Look at how the connective tissue is formed. This is dense, irregular connective tissue. Number 19, again, we're back to areolar. Look at how we have the bands and the nuclear, or not the bands, but the lines coming through the microscope, ocular lens, and look at how the nuclei are all over the place. Again, memorization. Number 20, this is look at how dense and look how compacted all these chondrocytes are. Okay, this is fiber cartilage, responds very well to stress. Pubic symphysis is a location as well as intervertebral discs. Okay, number 21, compact bone. Again, we have the osteon that's located here. We have the central canal. We have the lamella, the concentric lamella, as well as the osteocytes in the lacuna here. Okay. Oh, we'll talk more about this in chapter 6 as well. 23, we have hyaline cartilage. Okay, this is again chondrocytes. Look at their orientation. This is usually going to be found in endochondral ossification in the long bone, or as well as on articular cartilage. 24, very easy. Everyone should know this is blood. Okay, look at how the red dots has red blood cells. The purple stained dark are lymphocytes. Okay, they're slightly larger. Reticular connective tissue is number 25. Okay, again, orientation. Let's take a look at how the cells are oriented and everything. Here, we're talking about hyaline cartilage again, like we talked about before. Look at how the chondrocytes are, endochondral ossification, or articular cartilage. Fiber cartilage here, number 27. All right, 27 here, fiber cartilage. Look at how dense the orientation is of all the chondrocytes located within here. 28, okay, this should be compact bone. Okay, look at the osteon. Okay, we're going to talk more about this. It'll make more sense, but look at the circumferential, not circumferential, the lamellae wrapping around the central canal. Okay, epithelial tissue. This guy should be very easy based on what we just talked about. Okay, number 1, what we have here, we have simple columnar nuclei on one level, and they all look like columns. 2, we have transitional. Look at how the cells are bulbous, some are flat, all going to be responding to distension and stretch. We're coming through here and we're stretching out that bladder or that urinary tract. Okay, here, again, this is not adipose tissue. This is simple squamous. Look at how many cell nuclei there are wrapping around. Now, some of you guys get this confused with adipose because it looks like a giant blob of fat. But remember, nuclei, 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 one singular level of cells. Okay, here, number 4. We should all know this is simple columnar again. Nuclei are on the same level. 5, this is pseudostratified. Remember we talked about often ciliated. These are cilia on top, and the nuclei are all over the place. 6, we have multiple layers, 2 layers here, 2 layers of cuboidal cells, and this is a lumen stratified cuboidal. This is skin, everyone. Stratified squamous. Take a look. Okay, pause the video if you need to, but take a look at all the multiple flat plates that are being stacked right on top of one another. Right through here, number 8. Again, everyone not adipose tissue. This is simple squamous. Number 9, one single layer. Now these are not flat plates, these are cubes, so this is simple cuboidal. Number 10, okay, again, nuclei all over the place. We should all know this is pseudostratified columnar. And last one, stratified squamous. Here is the what you guys need to study. This is the key that is associated with everything we just went over. I should have gotten everything right, but we'll go over again this in class as well next week. So that's everything for this class. I want everyone to review it. You guys will have a test or a quiz based on just this lecture that I talked about through here, and we'll go from there. So thank you, everyone. Have a great weekend, and we'll see you on Tuesday.